Okay, now we're ready to get started. So thank you for coming. Um, really glad to finally be here to celebrate the accomplishments of Jacklyn Mohay, and like to introduce that our fiance James is here. Raise your hand so they can criticize you as well after the fact. And we want to hear today from Jacklyn Mohay. Woo! You did it to me. She gave me a margarita beforehand, so it's not my fault. She's been saying that to me for years. She's going to describe her thesis work about the spatial patterns in fish assemblages among benthic habitats in California. So we're really excited to hear her talk about her work because we spent many years literally at sea collecting the data, and she's taken the burden of analyzing incredible volume of video data. So that's really super. And Jackie graduated in 2010 with a degree in marine science and biology from Coastal Carolina University. And then for the next 6 years, she worked as a fishery observer in the North Pacific groundfish fishery, and also worked for the California Department of Fish and Game, working for a year or so studying the demographics, habitats, population, home ranges, and feeding habits of endangered and listed fauna. As you can imagine, working out of Fresno, she didn't have many marine animals in that list, but she did quite a bit of work in the field, and her work included habitat enhancements, radio telemetry, applying and removing radio collars, pit tagging, and collecting genetic samples from species that they trapped. So clearly, when Jackie came to us in the fall of 2017, she was a really experienced field biologist. And that was exciting to have her enter into my lab because I rely upon the older graduate students to teach the younger graduate students, and Jackie came in as an older graduate student and could start helping train the other students in the lab on a wide variety of issues. Given her experience, I was expecting that she would breeze through Moss Landing Marine Labs. But as it turned out, Jackie was a recipient of that famous saying, life is what happens when you're busy making plans. So it took her a little longer than most graduate students in Moss Landing Marine Lab. But good news is that when I first started working at Moss Landing, the average time to get a master's degree here was close to 10 years. So she's sped up from that. So that's a good sign. But here we are 8 years after she entered MLML, and I'm really impressed that she's here today and that she finished her degree because she's been working full-time for a couple years for applied marine sciences in Santa Cruz. And I have to say that I'm not surprised at that because one of her strongest characteristics is her strong work ethic. And I saw that she put her whole heart and soul into her work here. And so I'm not surprised that she was able to work full time and work on her thesis as well. It I'd like to have some of her energy, but she won't give it to me. So it was really impressive to see that. Now, I normally show you pictures of embarrassing photos. Take this back so I can get to the people on the Zoom call. Here is the time when I would normally show embarrassing photos of a candidate while talking about how good a scientist she is. So I'm not going to do that today. Not because I'm not willing to show embarrassing photos of Jackie, but because we have too many. There were too many to choose from. Instead of that, I'll just say that she has done a lot of things here at Moss Landing, and one of the first was to demonstrate her fish handling skills and her work at sea, as she was the volunteer coordinator for the California Collaborative Fisheries Research Program for a couple of years, and has spent a lot of time at sea for CCFRP. She also proved her seamanship by the many times a year that we spent at sea, and she spent at least a month a year at sea on our research projects. So keeping up with her academic life, her personal life, and her professional life while spending a month at sea was always difficult. But Jackie was always came through with a smile on that. She also learned how to weld here to get ready for going to sea. And she came up to me and said, well, I'd like to learn how to weld. And I said, well, how come? She says, well, I may not make it through here. So our great shop staff took the time to train her how to weld, and she was able to help us resolve some of our problems with our gear at sea through her welding skills. And importantly, she went from knowing nothing about geographic information systems to becoming our expert at that in our lab. And we use that intensively at sea as we integrated our navigational information from the boat, to our geographic information system, and also the video lander. So she basically on her own developed the skills to integrate all of that navigational information and put it into our GIS so that we could understand where we were sampling in the real world. And it was really, really helpful. She also survived COVID. And it was only once or twice when she was threatening to cut up the electronics in our video lander during COVID. But for the most part, she used her skills to fix our gear rather than break it. A couple times she probably broke it, but also she learned how to use the GIS for analyzing data. And our lab had a grant with the California Department of Fish and Wildlife to analyze the California halibut bycatch. And we did this on a spatial approach. And so she mapped a lot of information related to the catch and bycatch of California halibut. And we published a paper on that. And that information is still being used as they're still going through the process right now of trying to figure out what to do with the California halibut fishery. So your work really is continuing to help them. Also, she did all this while working as a bartender. And we would frequently take her up on her offer and use her skills as a bartender to get through the arduous field work. This, I believe, happens to be a picture taken from our Baja trip. And, you know, Scott's the reason why she's in an embarrassing position here. So, just in summary, Jackie has been an incredible friend to me now for 10 or 15 years, although at times I think she thought I wasn't a friend. And she's been a super teammate for our lab. We had a really strong affinity among all lab members, and everybody worked hard and worked well together. And she would always contribute her ideas, and it was always for the benefit of the group. And I really appreciated that, and almost all of our lab members were the same way, where we really worked together as a team. And Jackie was really instrumental in that, in getting people to say their opinions, but also to listen to other people as well. So it was really a pleasure to have her, and a pleasure to see her finish. So we're going to now hear from Jackie.
You really put a timeline on me there, Rick. That's not doing extend display.
Why is that not showing?
Display. Oh, okay. Thank you. All right. Well, again, thank you everyone for joining me here today. Really appreciate it. Rick, thank you for the very detailed intro. Yeah, thank you everyone. I'm Jacklyn Mohay. I appreciate you all coming to my talk. Today I'll be discussing my thesis on spatial patterns in fish assemblages across benthic habitats along the California coast. So avid fishers have come to learn that the type of species they want to fish for can be found in certain locations or habitats. For example, if you want to fish for rockfish, you have to find a rocky reef habitat, or if you want to catch halibut, you look in soft sandy bottoms. So we have this basic understanding of the distributions of many of these fishes, but I wanted to further understand why they are found in these certain habitats, known as fish habitat associations. So for my thesis, I used visual surveys to learn about these patterns and predict where we might see them. So why do these species habitat associations matter? Understanding the habitat associations of marine fish species is fundamental to marine ecology, conservation, and resource management. Complex habitats, such as rocky reefs, coral outcrops, kelp forests, and seagrass beds, support higher biodiversity and provide essential ecosystem services, such as providing refugia from predators and fishing pressure. So understanding these relationships allows scientists and managers to identify essential fish habitats, assess the impacts of habitat degradation, and identify critical habitats requiring protection. So if we wanted to explore the processes defining habitat associations, we have to consider other factors such as depth, salinity, temperature, which are all processes working at habitat levels to separate out these unique communities. So fish habitat associations are not static, and they often vary with latitude due to changes in oceanographic conditions, biogeography, and evolutionary history. Habitat composition, complexity, and spatial variation are widely recognized as key predictors of fish assemblage structure, species richness, and biodiversity. Species assemblages shift predictably with depth and habitat transitions, with rocky high relief habitats supporting more complex fish assemblages than unconsolidated soft sediments, and in contrast, these unconsolidated soft sediment habitats typically harbor benthic specialists adapted to less structurally complex environments. So all of these factors are important to fishes for reproduction, survival, and growth, unlike this green striped rockfish with zero survival instinct. So my goal for this study was to use data collected by Videolanders to provide a better understanding of these fish habitat associations and community composition variability along the California coast. So there are 554 fish species along the U.S. West Coast, 439 of which are found in depths less than 400 meters, of which those are a significant portion of our fishes are demersal species. For these demersal fish, habitat greatly influences their location. So demersal fishes, including economically important rockfishes, are often associated with benthic habitats that are structured by local topography and substrate type. So topography is defined as seabed complexity created by features like underwater mountains, trenches, and continental shelves. Substrate is material on the seafloor that forms the foundation of the benthic environment, such as rocks, sand, and mud. And relief is the vertical height of an object above seafloor, such as a boulder or pinnacle. And rugosity is how complex or rough a rock feature is. For this project, we defined it as the size of crevices or holes in a feature. So there are a number of different factors interacting to create habitat. Habitats are further characterized by varying substrates from hard rocky bottom to unconsolidated finer grained soft sediments, and range in seafloor complexity in the form of relief and rugosity. So the distribution and diversity of demersal fish communities are shaped by interactions among oceanography and habitat. A major network affecting habitats and the species within them is the California current system, which is one of the most productive and ecologically significant eastern boundary current systems in the world. It originates in the North Pacific Ocean and flows southward along the U.S. West Coast, transporting cold, nutrient-rich subarctic waters that fuel coastal upwelling and support diverse marine ecosystems. So this dynamic system influences physical, chemical, and biological processes from surface productivity to benthic community structure, making it an essential driver of marine biodiversity and fishery dynamics along the U.S. West Coast. So near point conception, the dashed line up on the screen, the CCS splits off, continuing to flow southward on the western side of the current, whereas the eastern side is characterized by the Southern California eddy, which is generated by the influence of coastline and topography. So Point Conception, located along the margin of the Santa Barbara Channel, marks a major biogeographic and oceanographic boundary between the Oregonian and Californian provinces, and represents a natural dividing line where cold, nutrient-rich waters to the north meet the warmer, more stratified waters of the south. So north of Point Conception, the continental shelf is narrow and steep, influenced by persistent upwelling in the cold California current, which promotes high benthic productivity and supports communities dominated by these cold temperate taxa, such as yellowtail, deacon, and black rockfishes. South of the point, ocean conditions shift under the influence of the Southern California countercurrent, resulting in weaker and more variable upwelling, warmer sea surface temperature, and increased water column stratification. So this transition corresponds with the shift in fish assemblages toward more warm, temperate, and subtropical species, such as California sheephead, blacksmith, and a suite of southern rockfishes. So the juxtaposition of these northern and southern processes at Point Conception provides a natural experiment for testing how demersal communities respond to differences in temperature, current structure, and habitat within a relatively small geographic area. So I focused my efforts on fishes that live on continental shelf and slope deeper than scuba depths and in untrawlable complex habitats where underwater instruments such as ROVs, manned submersibles, or AUVs are required to survey. Technological advances have revolutionized our understanding of fish habitat interactions beginning in the 1950s, where human-occupied submersibles offered some of the earliest in situ observations of fish behavior and habitat use, revealing strong species-habitat associations. Autonomous underwater vehicles and towed camera systems further expanded spatial coverage, integrating imagery with co-registered bathymetric and backscatter data to map benthic habitats and model fish distributions. These tools utilize transect sampling methods, which uses a path established along a steady area where data are collected systematically along that line, either continuously or at regular intervals. More recently, stationary or drop camera systems, such as baited remote underwater videos and stereo video landers, have emerged as cost-effective, non-invasive tools for rapid habitat characterization and species identification, particularly in untrawlable or sensitive areas. So these camera systems use a point count, which it consists of deployment to the seafloor, remaining in a fixed stationary location for a period while data are collected at this specific location. And thus, the stereo video landers were born. The rotating lander was designed and developed by Marine Applied Research and Exploration in 2012 to enable cost-effective, high-resolution monitoring of nearshore fish communities and habitats beyond the reach of traditional scuba or trawl surveys and comprehensive seafloor surveying. It featured a steel and plastic frame outfitted with 2 fixed-position, color, wide-angled, standard-definition video cameras that were on a rotating bar to provide 360-degree coverage. Video is transmitted up an umbilical for real-time viewing. So the rotating lander was deployed 459 times and recorded 1,331 drops. The Benthic Observation Survey System, or the BOSS, was designed and constructed in 2018 with the help of scientists and engineers from the Monterey Bay Aquarium Research Institute to be more rapidly deployable and lightweight with a hydrodynamic build. The BOSS is equipped with 4 vertically oriented paired cameras mounted internally for sampling and 1 downward-facing camera mounted externally. It also had 4 lights mounted on top of the lander and a downward-facing light mounted towards the bottom. The BOSS is mounted to a winch by a woven Kevlar tether that houses fiber optic and electrical cables that run to a topside recorder, which allows for real-time viewing. The BOSS was deployed 125 times and recorded 1,569 drops. So the BOSS and rotating landers were designed to speed up deployment and retrieval and allow serving a wider geographic span, particularly on high-relief rocky substrate. These landers were deployed in the water and hovered a safe distance off the seafloor to hop along a transect, taking multiple point samples. Deployments were counted each time the lander entered the water and ended when the landers were fully retrieved back on deck. A drop was constituted as a hop along a transect where the lander touched the bottom and a video was recorded. So each deployment consisted of multiple haphazardly located drops along a transect, usually 50 to 200 meters apart. So using data collected from the BOSS and rotating lander, I was interested in answering 3 questions for this study, including: do species richness, abundance, and diversity vary among habitat types, seafloor complexity, and depth? Do community assemblages vary among locations defined along the California coast, as well as the habitats within those locations? And can habitat type and seafloor complexity be predictors for fish size? So this study encompassed a latitudinal range of approximately 1,000 kilometers along the north central and southern California coast from Cordell Bank offshore San Francisco to San Clemente Island in the Southern California Bight. This region spans 2 major biogeographic provinces, the cold temperate Oregonian province and the warm temperate Californian province, as well as a transitional zone near Point Conception, which represents one of the most pronounced ecological breaks in the Eastern Pacific. Study sites were then selected to capture the dominant axes of environmental variation along this coastline, including latitude, habitat, and seafloor complexity. So moving on to my methods, our landers consisted of a camera configuration that included a pair of color, wide-angled, standard definition video cameras mounted obliquely and equipped with 2 dimmable LED lights. So for those of you who may not be as familiar with stereo video, it just means the landers consisted of 2 cameras mounted vertically, towed in 7.5 degrees to provide adequate camera overlap for stereo analysis. And so this allows for more accurate measurement of fishes caught in both cameras. So how did we extract data from the rotating lander? Species accumulation curves calculated from rotating lander data were used to determine that 8 rotations were sufficient to capture the majority of species richness and relative abundance at a given site. Stereo video projects were created in Event Measure software by importing paired video files, calibration files, and species attribute lists. Fish were enumerated using a modified max-n approach, where the maximum number of individuals of each species observed during a single 360-degree rotation was recorded. So this modification, which differs from traditional frame-based max-n method, minimized double counting while maximizing detection potential. So for each fish included in max-n counts, total length was measured in 3D by marking the snout and caudal fin in both the left and right video frames. Z distance, or the distance from the camera to the fish, was calculated for each individual as well. So to account for species-specific detection limits, the affected area surveyed for each species was estimated using a radius equal to the 95th percentile Z distance observed in all deployments as defined in Starr et al. 2016. So this radius, along with the predefined minimum detection radius, was used to calculate the effective area sampled as a donut-shaped annulus. And this approach accounted for differences in detectability related to species morphology, behavior, and environmental conditions. Fish densities were then calculated as a species-specific max-n divided by the effective area surveyed. So BOS data were extracted in the same way. Once surveys were completed, the stored videos were transcoded and downloaded from the deck recorder. Individual files were then synced to a single composite video using VidComp software from each of the 8 sampling cameras, excluding the downward-facing camera. So videos were synced using a light flash conducted at the start of each drop, and then from this point, they were stitched together using the same timestamp and paired with the calibration files. Videos were then analyzed using event measure software where fishes were enumerated and identified to the lowest taxonomic level possible, and a max-n frame was chosen for the total count of a single species in each video. 3D points were assigned to each fish during the species-specific max-n frame to be used to calculate densities along with the survey area determined using the lander-specific calculations. So here in this figure, you can see there's a 24-degree gap in the field of view between each of the 4 camera pairs, and that's to prevent double counting of fishes. So this is an example of a max-n frame chosen for blue rockfish. Here you can see that they were enumerated and identified to the lowest taxonomic level possible, and each max-n frame is species-specific. Total lengths were then estimated using 3D points at the max-n frame, although this only consisted of a subset of fish, as only fish with acceptable camera orientations and those that were visible in both camera frames were measured. So total lengths were measured by zooming into the fish at 4 to 8 times magnification and measuring from the tip of the snout to the edge of the fin membrane in the center of the caudal fin. These lengths were referred to as total lengths because the fin membrane was often blurred and the tip of the caudal fin was estimated on best judgment. So previous studies looked at differences among sediment types and microhabitats, but we found that these relationships stand out better if you use higher level categories. So during surveys, in situ habitat classifications of mud, sand, cobble, boulder, or rock ridge were assigned to each drop. And then during video analysis, a habitat code of hard, soft, or mixed soft was assigned to each drop after corroborating the in situ classification. So hard bottom type consisted of no soft substrate, such as rock ridges and boulders. Soft consisted of unconsolidated sediments, such as sand, silt, and mud. Mixed hard was defined as any mixed hard and soft habitat that contained 50% or greater rock or boulders or cobble. And mixed soft was defined as any mixed hard and soft habitat that contained 50% or greater mud or sand. So we started categorizing vertical relief and rugosity as variables in video analyses starting in 2019, so only a subset of later data included these. Each parameter was divided into 3 categories, high, medium, and low, and a drop was categorized as high relief if the tallest point in the frame was 2 meters or greater off the seafloor, medium if the tallest point was 1 to 2 meters off the seafloor, and low relief if the feature was 0 to 1 meter off the seafloor. So regardless of the number of holes or crevices, drops were categorized as highly rugose if a crevice or hole is large enough for a large fish, such as a cowcod or a large lingcod to enter. Medium if a crevice or hole was large enough for a small rockfish, such as a square spot or rosy to enter. And low rugosity if there were no crevices or holes present. So we kind of see a pattern between soft bottom, low relief, and low rugosity here. So data sets from each tool were combined and standardized to only include unbaited and environmentally comparable drops for a combined total of 1,117 drops and 147 deployments. Matthews et al. 2024 determined there were no sampling bias between the 2 video lander systems So this, along with scientifically standardized sampling protocol, allowed for the two data sets to be combined. Drops were further grouped into locations based on proximity, where each tool collected data at similar depths and habitat types in bioregions either north or south of Point Conception. Drops targeted depths ranging from 50 to 200 meters, and deployment and drop sites were selected to target rocky substrate with adjacent soft bottom habitats. So to evaluate patterns of fish assemblages and habitat associations, I calculated several response variables describing community structure across all habitat types, depth, sampling locations, and bioregions. These included species richness, abundance, and diversity. All observations were assigned to habitat type categories, and a subset of drops were assigned relief and rugosity. I then assigned drops to locations distributed along the California coast. Locations were further grouped into bioregions north and south of Point Conception, and drops were categorized into 30-meter bins to reduce data noise and provide a more even distribution of samples. Fish lengths were also standardized across species, dividing the total length by the species length at 100% maturity. So to test for differences in community metrics across habitat types, locations, depth, and bioregions, I used Welch's ANOVAs. Prior to analysis, assumption of normality and homogeneity of variance were evaluated, so I then used post hoc pairwise comparisons using Games-Howell tests for non-parametric results. Species were then categorized in family groups and various taxonomic groupings to look for patterns in density across locations and habitats. I used generalized linear models with the factors of location, bioregion, habitat, relief, rugosity, and depth. And then I ran a nested ANOVA between bioregions with locations nested within these bioregions to test whether species richness, density, and diversity differed between the regions or among the locations within the regions. To assess whether overall community structure varied across the California coast and between bioregions, I ran a distance-based redundancy analysis to examine whether habitat or site explains more of the variance in community structure, followed by a permanova to test how those factors affected it. Species abundances were square root transformed, which reduced the influence of highly abundant taxa and allowed comparisons of community structure across scales. So then to evaluate how physical habitat characteristics influence fish body size, I conducted Welch's ANOVAs to test for significant differences in mean fish length among habitat types, relief, and rugosity levels. I standardized fish length to size at 100% maturity using length at maturity tables for among species analyses, and I plotted average fish lengths for the most commonly observed species across different structural complexities. I visualized these relationships using violin plots, which illustrated the distribution and variability among species and within species to these metrics. So, in total, we observed 29,975 fishes from 115 unique species between offshore San Francisco to the southern Channel Islands. Fish communities were numerically dominated by rockfish species across all locations. Lingcod, yellowtail rockfish, and copper rockfish were the most abundant in northern and central Californian sites, and the more southern sites were occupied with black-eyed gobies, olive rockfish, California sheephead, and ocean whitefish. So hard and mixed hard habitats most commonly consisted of species with affinity to higher relief and rocky habitats, such as vermilion rockfish, square spot rockfish, and Sebastimus, which is a subgenus of Sebastes, consisting of rosy rockfish and starry rockfish and similar species. Mixed soft habitats showed a species change to primarily half-banded and green-striped rockfish. And in soft habitats, we saw mainly flatfishes and sandy bottom associated rockfishes, such as half-banded and green-striped. So I found species richness varies by habitat and depth. Hard and mixed hard habitats showed the greatest species richness among the four categories, with soft showing the least and mixed soft generally falling in between. Richness increased with depth across all depth categories, with significant differences occurring between shallower, intermediate, and deeper depths. And species richness also varied by location. So I found Big Sur contained more species-rich drops than other areas, and this may be due to its location on the boundary of two bioregions and its distance from port, reflecting lighter fishing pressures. Big Sur also had the highest proportion of hard substrate that we sampled. Species richness was also highest at Santa Barbara and the Southern Channel Islands, and the Southern Channel Islands had the highest relief and rugosity of the sites that we sampled. So density varied across habitat and depth as well. Across habitats, mixed hard and mixed soft habitats showed the greatest density, followed by hard and then soft with the lowest. However, we're targeting a mixture of habitats near rocky reefs, so it's possible we'd see an increase in abundance of soft habitats if we surveyed closer out to the middle of the open ocean. So overall density declined with depth. Shallow to mid-depth categories had higher values than the deepest, with the greatest densities between 111 to 140 meters. Category 1, from 20 to 50 meters, only included 7 drops, so the standard error from that's pretty large. Densities significantly differed among locations as well. Densities were highest in the Northern Channel Islands, Santa Barbara, Southern Channel Islands, and Big Sur, and lowest in Monterey Bay, Morro Bay, and offshore San Francisco. So pairwise comparisons show that the southern sites had greater fish densities compared to northern sites except for Big Sur. Again, this is most likely due to its location on the edge of the bioregional divide, where warm and temperate species overlap, and there's a greater distance from fishing ports and proportions of hard habitat. Morro Bay may also show low densities because while we were sampling there, the currents were pretty strong. Density analyses were further analyzed by family and taxonomic groups. Overall, the figures indicate a broadly consistent rockfish-dominated fauna that becomes more compositionally diverse in the Southern Channel Islands and on soft substrate where non-rocky reef and warmer water-associated species are present. Shannon-Weiner Diversity Index varied significantly across habitat. For each group, the box spans the interquartile range of H'. The horizontal line inside the box is the median, and the whiskers represent the more extreme observations. Inside each box is the mean plus or minus 1 standard error. So hard and mixed hard habitats supported greater diversity than soft substrates, with mixed soft generally intermediate. Diversity moderately changed with depth. As you can see here, diversity generally declines with depth, where softer habitats are often associated at deeper depths. Big Sur showed the highest species diversity compared to other locations. The Southern Channel Islands were more diverse and hosted a greater suite of species of all the southern sites. And Morro Bay showed the lowest diversity, potentially due to the narrow range of depths sampled at that location. So I used mixed effects nested ANOVA with bioregion as a fixed effect and location nested within bioregions to test if species richness, density, and diversity varied between bioregions. I found that only fish density was significantly higher in the south than the north, indicating a clear biogeographic shift in overall abundance. So overall, these patterns corroborate with previous findings that species densities increase equatorward and that the Channel Islands MPA network and distance from mainland may influence fish densities. GLMs show that species richness, abundance, and diversity all varied significantly among habitat depth and location, but only species abundance varied by bioregion, and only species richness and diversity varied among relief and rugosity. So I then ran a distance-based redundancy analysis in Permanova to see how community structure changed among habitats and locations. So distance-based redundancy analysis is a multivariate statistical technique that uses a 2-step process to explain community composition based on environmental variables. It first calculates a dissimilarity matrix to the community data, and then uses a principal coordinate analysis to convert the dissimilarities into a set of coordinates. A redundancy analysis is performed on these coordinates to test the significance of the environmental variable's effect on the community structure. So in this plot, the colors represent different habitats and the shapes represent locations. So my DBRDA showed that both habitat and location influence community composition. The arrow directions indicate the gradient along which each species' relative abundance increases, and arrow length reflects the strength of that association. So here you can see sites cluster by habitat types with modest overlap, suggesting that habitat differences contribute to structural assemblages. Species vectors show that square spot rockfish are the most strongly associated with hard and mixed hard habitat, along with yellowtail, vermilion, pygmy rockfishes, and ocean whitefish. Soft habitats are more strongly associated with smaller schooling species, such as anchovies and half-banded rockfishes. So overall, there's a clear habitat separation, primarily driven by these smaller schooling species. The second DBRDA output shows location by color and habitat by shape. So this plot illustrates a clear differentiation in fish community composition among geographic locations, especially distinguishing northern sites from the southern sites. Vectors show fish assemblages that strongly influence these patterns, including vermilion, sebastimus, and yellowtail rockfish, are primarily associated with northern sites, and black-eyed goby, ocean whitefish, and half-banded rockfish are strongly characterized by the southern locations. The distinction between clusters emphasizes the ecological uniqueness of these regions by location and habitat, and although we see significant clustering, there is some overlap among location groups, which indicates a shared species or transitional ecological condition, underscoring the complexity of ecological interactions and community structure along latitudinal gradients. So habitat type, complexity variables were categorized for a subset of drops and compared to average fish lengths, standardized to length at 100% maturity for each species to see if habitat, relief, and rugosity could be a predictor for fish size. So standardizing fish length at 100% maturity allows for the comparison across species by removing the confounding factor of growth rate differences, providing a standardized measure of reproductive potential relative to body size. So I found that habitat was a significant predictor variable for fish size, with larger, more robust species commonly found in harder substrates. When I looked into these on a species-specific level, I found that hard and mixed hard habitats generally supported larger-bodied individuals such as lingcod, copper rockfish, and flag rockfish. Short-belly rockfish and half-banded rockfish were more uniformly distributed in length across all habitats, suggesting lower habitat-driven size selectivity. So longer-tailed distributions in the hard habitats were observed for species like canary rockfish and vermilion rockfish, indicating habitat-specific growth potential, aggregation of older individuals, or species that experience these ontogenetic shifts in habitat use. Similarly, several species showed smaller median body sizes in soft compared to hard habitats, such as lingcod and chili pepper rockfish. So the ultimate take-home message here is that violins with longer tails in hard and mixed hard are driven by these larger species, and that differentiations in soft and mixed soft violins are driven by species such as lingcod and chili pepper. So larger fish equals more reproductive potential. I found that high relief areas consistently supported larger bodied individuals for a broad range of species, including cowcod and lingcod. These patterns highlight the role of vertical structure and complexity in offering refuge, food availability, or reduced predation, which are key factors in supporting larger fish. So some species, such as blue rockfish, appeared more uniformly distributed across relief types, indicating that there's more generalist and exhibit weaker associations with relief-driven size differences. Chili pepper and widow rockfish tended to be smaller in low relief areas, suggesting seafloor complexity may influence growth or habitat selection. So there's not as prominent of a difference among these violins, probably driven by very few species. And blues may not have such an association with the higher relief as they are more semi-pelagic species as well. So lastly, across species, body size changed only modestly with rugosity, but fish tended to be larger in rougher, more complex habitats. Chili pepper rockfish and widow rockfish again showed the greatest variability in size compared to rugosity, while lingcod, green-spotted, vermilion, and canary rockfishes varied little. So comparing fish lengths to relief, rugosity, and habitat can help visualize how physical complexity filters both species presence and their size spectrum. And by doing so, fishery managers can spot which microhabitats harbor larger, likely mature fish that sustain fisheries, guiding spatial protections. So by investigating differences in rocky reef complexes with unconsolidated soft sediments adjacent along a latitudinal scale, my study confirms that habitat type, complexity, depth, and location all affect species richness, density, and diversity. I also found that habitat and seafloor complexity are linked to fish sizes as species experience ontogenetic shifts and aggregate in more complex habitats as older individuals, although this could also be species specific. I also found hard and mixed hard habitats provide more refugia, foraging opportunities, and niche partitioning. So my research shows that habitat impacts fish communities on smaller scales, but there are biogeographic and oceanographic drivers acting on these larger scales. Species richness differences across point conception reflect water mass regimes and increased richness of fish toward the equator, as was found in Roboski et al. 2018. Spatial variations in habitat heterogeneity and oceanographic regimes also significantly influence fish population densities across marine bioregions. So finding community assemblage differences between habitats and locations were often driven by these small schooling fish like square spot and half-banded, means a relatively small number of key species could be used to efficiently track changes in fish communities for monitoring and management. Soft, sandy, or muddy seafloors tend to have their own set of species, which are quite different from those living in more complex rocky habitats and around places such as Big Sur and offshore San Francisco. So the highest diversity of rockfishes along the California coast is found in Southern California Bight, where the mixing of cooler and warmer waters creates especially favorable conditions, and there are range overlaps of temperate and subtropical species. So overall, this pattern shows that fish communities are divided into regions, likely influenced by ocean features, how productive each area is, and other environmental factors. However, as ocean changes with climate change, we may see ecosystems reorganize, with more subtropical species moving north and replacing species that prefer cooler waters. Areas with flat, simple seafloor offer fewer hiding places and are more easily damaged by fishing gear that drags along the bottom, which can lower survival of juvenile fish. In contrast, rocky reefs create more complex habitat that give fish shelter and can even make fishing less effective as gear snags more easily. So the Northern Channel Islands, Southern Channel Islands, and Santa Barbara had the highest fish densities, likely because these areas have a mix of different habitats, some protected from marine protected areas, and are farther from mainland. And a lot of these included schooling species such as blacksmith and ocean whitefish. Differences in depth and latitude may also be tied to how much rocky versus soft bottom we sampled in each area, and the schooling midwater species can strongly affect how many fish we see in a given place and habitat. So more complex seafloor structure may also help concentrate pelagic prey like krill by influencing local water movement. So species richness is a function of habitat and depth. At a local scale, the number of species tend to be higher in places with more structure and uneven seafloor because these areas offer shelter, feeding spots, and different living spaces than flat, uniform, sandy, or muddy bottoms don't provide. Past studies have shown that fishing gear that drags along bottom can make these soft bottom habitats even poorer by flattening the seafloor, removing living structures like corals or sponges, and generally simplifying the habitat. Other research has found that seafloor structure becomes more complex, and predators can't see or reach the prey as easily, which further highlights how important seafloor structure is to refuge. So the main break between different groups of marine species is near point conception, but recent ocean warming has increased the number of species in central California as more animals shift their ranges north. So in the past, island fish communities have been shown to cluster in warm water and cold water groups that don't always match the simple geographic distance. Some species follow the expected patterns, but others do not, and this shows that habitat type, the broader regional setting, and impacts of fishing all work together to shape where fish are and how many species occur in an area. Higher diversity in hard bottom habitats suggests that physical complexity plays a key role in sustaining species richness and evenness. Higher diversity in Big Sur and the Southern Channel Islands suggests even more species distributions influenced by temperate and subtropical species influxes. Likewise, prior studies have identified higher diversity in rock reefs and offshore soft bottom habitats in Southern California, attributing these patterns to a variety of available habitat types and the influence of warm water masses supporting both temperate and subtropical fish species. Diversity in Southern California may also be influenced with the western side of the Channel Islands experiencing effects of the cold California current. These patterns may be driven by latitudinal gradients in species composition, where tropical and subtropical species influence the community dynamics in the south, promoting more even assemblages. Similar findings have been reported in studies of benthic fish communities across broader geographic ranges. So for many reef fish, bigger individuals are more often found in complex, high relief habitats. So loss or degradation of high relief complex habitat is likely to disproportionately reduce the abundance of larger individuals, with potential consequences for reproduction, as bigger fish usually produce more eggs, predation and trophic structure, as larger fish play different ecological roles, and in fisheries, as bigger trophy fish are often targeted. So hard and mixed hard bottom areas tended to have larger fish, probably because they offer better feeding and protection, and because some species move into these habitats as they grow in these ontogenetic shifts. On the other hand, species that don't show big size differences between habitats may be less affected in terms of size structure by changes at this broad habitat scale. However, they may still rely on specific microhabitats that are not fully captured in the broad categories. So overall, these patterns have important implications for how we understand ecosystems and how we manage fisheries and habitat protections. So what does this all mean for monitoring implications and future direction? Given that community structure align more strongly with geography included than with local habitat alone, future studies could benefit from integrating oceanographic data and larval dispersal models to better elucidate the drivers of spatial community dynamics. Additionally, incorporating behavioral metrics or dial comparisons might reveal more about a function of roles of different habitat structure. Artificial intelligence offers a promising tool to accelerate analyses by automating species identification, tracking, and length estimation from stereo videolander data. Furthermore, the spatial and biological data sets generated in this study could help train machine learning models aimed at predicting species habitat associations at scale, ultimately supporting ecosystem-based management. While stereo videolanders were validated as effective tools for broad-scale community assessments, further refinements in deployment strategy, video resolution, and integration with other observational platforms could improve estimates of size structure and better detect fine-scale associations. And next steps for videolanders could include incorporating oceanographic sensors to the landers for in-situ oceanographic data collection alongside biogeographic surveys. So, first and foremost, I'd like to thank Rick for teaching me the ethics of work hard, play harder. Take frequent naps when necessary, even if we're on the clock. And that it's okay to be a lifetime student. But in all seriousness, I am forever grateful for your role in shaping me as a scientist who I am today. I could not have made it this far without you, especially all the late hours and weekends that you've sacrificed helping me wrap this up. So thank you. This picture was taken one day before I joined his lab. Scott, I'm forever grateful for your guidance through all my classes and time at Moss Landing. You've challenged me in my statistical approach more than I could have imagined. I've probably learned more in the last few months than I have in any stats class, although we'll talk about that later. Maybe I didn't learn. All right. James, you employed me as a TA since day one, even though I'm not sure how qualified I am to fill that position. But I am grateful for your advice and helping me develop my first thesis and talking out concepts with me and always being there. Thank you. And Ivano, I appreciate your guidance during my time here at Moss Landing as well. I'll never forget our geo-ocean trips, fossil digging, and learning about the formations that make up the Bay Area, and all the time we spent learning how to get the M3 multi-beam sonar to work. That actually got me into GIS, so thank you. To the FCB lab, Katie, I see you over there. I could not have done this without all of you guys. I appreciate your continued support, even as many of you have moved on in your careers. Yeah, Jake, thank you for setting up the room today and all of your help with that. Katie and Kinsey for sitting in on my practice talks and everybody else who offered. Faculty and staff in Moss Landing community, especially Tara for always being there, checking in and answering my arguably stupid questions. Everybody in facilities and in marine ops, greatly appreciate all of your help and time. Mbari and Mare scientists and engineers who created these landers, my work would not have been possible without you. Captains and crew of the Fulmar, Shearwater, and Outer Limits, thank you for putting up with us and keeping us safe on the water. AMS, thank you for allowing me time to work on my thesis and for keeping the fire lit by asking when I'll be done. To my friends and family, many of you who flew in today or watching online, thank you for your love and support throughout this extremely long journey. And last but certainly not least, James, my fiance and partner in crime. I've officially been a student for 80% of our relationship. So thank you for your patience while I work long hours to wrap this up and for telling me to go work on my thesis when I just wanted to annoy you. Imagine what I'm going to do with all my free time now. And with that, I'll take any questions.
Give her a hard time on.
All right.
Katie?
Hey, Jackie. Oh, okay. Hey, Jackie. Great talk.
Thank you.
Thanks so much. My question for you is, what is the weirdest thing you've ever seen on a fish video? I want to specify fish video in there.
Okay, I was like, wow, these opportunities are endless here. The weirdest thing, well, probably the fish behaviors, to be honest. It's alarming how stupid some of these fish are. I know we have seen some vermilions just swim right into rocks as they get startled. And then I think there was one video where a lingcod was trying to shove a whole salmon into its mouth sideways. That was pretty interesting. Yeah. Thank you.
I thought you were going to say when we put the video lander down on those whales in the Santa Barbara Channel.
Oh, yeah, that was pretty interesting. That was good. Yeah, we didn't touch them, guys. We didn't touch the whales.
Great job,
Hey,
Jackie.
Amanda. Thank you.
Hi. Great job. That was really cool to see. I would often get messages from Jackie being like, hey, what's this cool invert that I see? And so this is what I know you didn't document them or catalog them, but I am curious whether you observed or felt like there were any associations that were more for the biogenic habitat rather than like the hard substrate. So like within a category of hard substrate, what did you see for?
Yeah, within a category of hard substrate, a lot of Metridium, I would say. I'm trying to recall if there was any specific species that we would see hanging around the Metridium fields. I picture a lot of vermilions. I think I had one picture that had a bunch of Metridium in it. But yeah, we did that whole corals cruise that we did up here in the Monterey Bay, a lot of cup corals, but unfortunately we didn't really document any associations between those, but that would be interesting for future study.
Yeah, I'd be curious. Fish, sometimes when we were working in the sponge reefs, the hake would come down and then they would bite the tips off of all the sponges. So, I mean, I'm sure there are things that they like to do around the inverts around there.
Somebody else had a question up here, or was it you, Scott? Sure. Oh, no.
I was looking at your size results in those figures, right? The body size results. And in general, the fish were smaller in the low relief and low rugosity habitats. But there was an interesting kind of like bulge at the largest sizes. There were like a lot more kind of big, relatively big fish also in that habitat. So was that potential predators that are hanging out in that area? Or was that just they're small fishes, but they just happen to be the largest of the small fishes?
So in the low relief and low rugosity, there were larger
there was
fishes
like
as well.
extra,
We can go back to those
kind of
plots.
Popped up again.
I'll go all the way back. You know what? Save us some time here. Okay. Yeah, so in the low rugosity,
Right, yeah, they're mostly smaller, but then you see that kind of tail
Yeah.
in the larger sizes. So I'm just curious, is
Yeah.
that just, these were just the biggest of the little guys, or were these are the predators that like to hang out and eat the little guys?
yeah, it could be predators. It also could be the fishes that experience these ontogenetic shifts if they're looking to, go lay eggs in these higher relief, higher rugosity areas for protection. It could be that as well.
I'm surprised you forgot. We talked about this a week ago.
Oh, that anything over one is,
Well, I asked her the same question, a week or two ago. And, one of the things that we can account for in this approach is shifts based upon movements of different species related to reproduction. So we think that some of that in the low relief soft sediment habitat is the occurrence of large lingcod that are moving through an area, and you would expect large lingcod to be living on rocky substrates, but we occasionally see them out on soft substrates, perhaps as they're hunting. So that's an interesting question, but reflects some of the challenges of interpreting this kind of information.
Who are those species that are in that tail?
That's right, that's right. Other questions? Oh, boy, you're getting off easy. Well, the good news is that, one more.
So your data for the densities, right? In general, there were higher densities in Southern California.
Yes.
What if you looked at biomass? Do you think you would see also more biomass, or would it shift because there's bigger fish in general in Northern California?
I would think it might shift because we saw in the DBRDA there was that clear structural difference from half-banded rockfish, and so I wonder if that would make it shift, and that and the blacksmith as well are fairly smaller bodied fish. So I would imagine there might be a shift. That'd be interesting to look into.
Ooh, that's fun.
Okay.
Yeah, there you go.
Sponson, I'll be right back to you.
That was awesome, Jackie. It's really cool to see, all those videos, and the talk was great and good work.
Thank you.
My question is about a little bit more about habitat, and maybe you could give like an anecdotal answer to this, but I was curious in thinking about your metrics of relief and rugosity, so relief being vertical distance from the seafloor and rugosity being the size of the holes, correct? If you observed or maybe quantified in some way whether there were size or species-specific associations with different levels of relief or rugosity, like perhaps at lower rugosities there would be more smaller bodied fish or small individuals or something of the like. If you could speak to that, that would be awesome. Thank you.
Yeah. Anecdotally, I would say that the smaller, like lower relief, lower rugosities, because the smaller sized fish could fit into these holes and crevices and hide out, they would be more associated with those habitats, but we didn't really quantify it unless that was something that you guys did after I moved on. But yeah, we never looked at size of fish specific to the different release and rugosity, but I'm sure we have that data in the data set we could probably look back at.
Nice work.
Thank you.
I think one of the things you talked about up front in the introduction was that you didn't really sample unconsolidated sediments per se, but more adjacent to rocks.
Yep.
Do you have any sense of what the community would look like using this tool if you moved away from the reef, like out into truly unconsolidated sediment of the continental shelf?
I imagine it would shift to mostly flatfish rather than seeing these green stripe rockfish and these, more rocky adjacent rockfishes. We would probably see a lot more flatfishes, maybe some rays. Yeah.
That was an opportunity to say this question goes beyond the scope of my thesis.
Oh, that was a great answer.
I was just trying that out.
Black-eyed gobies.
Okay. Any other
Dabble.
questions? Well, we're going to take Jackie into this small, hot, enclosed room and grill her for the next 45 hours. But please feel free to hang out and eat and drink, and she'll be out in a few minutes. Thanks very much. One more.
Thank you.